Hello, my name is Michael Edwards. As Enrique said, I got really sick of writing grants, didn't get funded. So I decided to move out on my own, and I've been doing data analytics for about 3 years now. Not only do I look at biological systems, but I'll look at anything with numbers. And in fact, recently I analyzed survey data from an entertainment venue. They put out a survey for 2,000 people. They got all the results back and no one knew what to do with the data. So somebody knew me, and so I've been analyzing survey data for them. This isn't going to be your typical talk. This is going to be very kind of, informal. That's kind of how I am. I do not have a lot of computer skills or mathematical background. That when I first started doing this is about when microarrays came out. Everybody heard of microarrays? Affymetrix, maybe? No? Oh my gosh. I do have some gene chips. I'll pass them out later. But anyway, basically it gives us the ability to measure all transcription at the same time. And I thought that was pretty awesome because I was on, I think it was like my 15th RT-PCR for the day, and I was like, this is terrible. I can look at everything at the same time. Why do I do all these, RT-PCRs? So basically, I acquired the mathematical and computer skills that I would need to basically do this. And also what I'm going to demonstrate to you today is some of the software that I use. I do not program, and that is like forbidden in bioinformatics. Everybody, like, I can't believe you don't program. It's like, I don't want to talk to the computer. Computer's boring. I want tools that allow me to I'm a biologist at heart, and actually, oh, come on. Okay.
Computer's mad at you.
I know, geez. So I kind of look at all these things, all this data out there as kind of pixels, right? And if you look at your average big screen TV, actually 4K has probably got about 8 million pixels in it, or boxes, right? When you watch your TV, do you see all those boxes? No, of course not, right? Your mind puts this all together and makes a picture, right? And these pictures can make you laugh and they can make you cry, but in essence, all you're watching are boxes, right? And in essence, when we analyze biological information, all we're analyzing, its very simplest form is zeros and ones, right? How do I take all this information and put it together in a picture that people can understand, right? That my collaborator can basically look at this information and say, aha, we should go after this gene because of this picture. So today, in today's talk, I've got lots of lectures. Please, like I said, please go check out my YouTube channel several times. But my big thing is I like to use other people's data. And if you talk to a lot of established scientists, they're totally against that. That there is a lot of information out there. There's way more information than you could possibly generate yourself, right? And everybody thinks that their project's very, special, and I work on this and no one else works on this. You'd be surprised. You really would. Especially in cancer data. There's so much data out there. So what I'm going to do is, in this talk is I'm going to introduce you to the wisdom of crowds. And this is basically kind of the theory behind why we can use all this information and get something that's better than each individual piece. So what I'm going to show you today is that we can use all this information and build a study that's way better than anything anybody else out there has actually built. So I'm going to talk about kind of the history, some of the uses of it, and then some of the conditions of wisdom of crowd and how that applies to biological information. Then I'm going to give you some specific examples of myself using the crowds in biological research. And in the first one, I'm going to talk about collecting information to investigate a phenotype. In this case, it'll be a disease and a cancer type. My second demonstration will be, how, we can use this information to investigate a disease, but how can we use it to, like, say you're just interested in a specific gene? How can you use this information to look at, what is this gene doing and how can we modulate it? And then at the very end, I'll take questions. So before I get into all this high-tech stuff, I got to talk about a story about 120 years ago. And this was a contest at a county fair in England, and it was basically a raffle. Well, it wasn't a raffle. People they showed an ox, and basically everybody had to guess how much the processed meat from the ox would weigh. the person closest to the guess got all that meat. And I don't know about 19th century, England, but I'm sure that was a lot of meat. That was a big prize. So you had to pay some money and people put in guesses. Well, at this county fair, there was this guy named Sir Francis Galton. Anybody hear of Sir Francis Galton? You're not included, Enrique. I know I've told you, you've heard this talk. I know. I talk about him a lot. Anybody heard of Sir Francis Galton? He was Charles Darwin's cousin. I think he was actually smarter than his cousin, but he was also known as the father of eugenics, which is probably why history's buried him. Everybody know what eugenics is? Okay, good. Well, what's funny though is this whole wisdom of crowds kind of, disputes that whole, eugenics theory. So he saw this contest and he thought this was a very interesting way to just kind of see, test his eugenics. He had a few people he knew that were in the livestock industry that should know how much a ox weighs. So he had his experts, and then he had basically the rest of everybody guessing, right? So again, we're guessing the processed meat of an ox. About 800 people paid to guess the weight of the ox. They had to throw, I think, 13 of the guesses out because they couldn't read it. But again, you have your experts, which are small, and your lay people, which are large, right? Again, he thinks his experts are going to be way more accurate than the lay people. So he goes to the people running the contest, like, hey, can I see the results? And by this point, he was knighted. So I think the contest people are like, yeah, whatever you want. I don't care. You're Charles Darwin's cousin. All right, whatever. So he basically looked at all these guesses, and what he realized is that no one got the exact weight of the bull, right? Out of all these almost 800 guesses. But here was his aha moment, is that when he took the average of everybody's guess, it was in 1% of the actual weight of the bull, right? You can kind of picture this as, say we put a dartboard on this wall here, and I had all of you throw darts at it, right? No one hits the dartboard, but if I take the geometric mean of all your throws, it's probably pretty damn close to the bullseye. We can do the same thing in biology, I swear. So he was amazed. He's like, oh my gosh, I can't believe that stuff worked. He's like, I really thought my experts were going to be way more accurate. But it turns out when you take everybody's guess and take the average of that, it's way more accurate than anybody, right? And he also said, I think we should study this more, for sure. He published it in Nature in 1907. He actually titled it Vox Populi, which is Latin for voice of the people. I think if I ever have a rock band, I'm going to name myself Vox Populi. Actually, there is a rock band out there, and they're not very good, named Vox Populi. Hopefully none of you are Vox Populi fans. Okay. So, and I mean, it's less than 2 pages. You can actually go and look this stuff up, right? Those were the good old days, The structure of DNA was a page and a half. How long was your last page, the manuscript that we were on? 10, 12? Yeah, plus supplemental. All right. So ever since, Francis Galton, actually they've been using this in history, but he's the one that pretty much popularized it, is that we've been using the crowd to predict the future. There was a recent book out there, James Surowiecki. It was called The Wisdom of Crowds. Does anybody read that? You should. It's on the New York Times. I think it was on the New York Times bestseller list for a while. But anyway, he goes into all the uses and the different ways people are using this. How about Nate Silver? Does anybody know Nate Silver? The website 538, does that ring a bell? Yes. Cool. Well, he used wisdom of crowds in his research. And he actually predicted almost every major election in this country in 2012. Do you think he asked anybody who they were voting for? No, he did not, right? All he did was take all the polls that were out there, right? Some were obviously leaning left, by the Democrats. Some were leaning right by the Republicans. But when he took the average of all those polls, he got the results of the election. And it's not only him. Our government's using wisdom of crowds. So the CIA is actually taking groups of lay people and asking them geopolitical questions like, what's the probability of Russia invading Ukraine in the next 3 months, right? And all these people give these guesses. They take the average of those guesses. Is somebody from Ukraine? Yeah. I hope they don't. Mine's 0%. They are actually, yeah. So they'll ask them, and then they'll take the average of all these people, and their guesses will be better. The average of all these people will be better than the guesses of the CIA agents that they've had for 30 years, right? There is something to this, that the more people looking at a problem, the better it is you're able to solve it. Okay? But there are 4 requirements. Diversity, you can't ask the same type of people. The more diverse the crowd, the better, and that works for research, right? All these labs are competing against each other. They don't want to replicate everybody else's stuff. They come from different backgrounds. It works perfectly in biology. Again, independence, that you can't influence somebody's decision, right? And sometimes in biology, other groups can influence other people's research, but usually when you dig into the raw data, you get exactly, you get not their interpretation, but kind of the truth out of it. Specialization, people are very knowledgeable in specific fields. Again, this really works in biology. You can have 2 different groups working on the same cancer, but one might be immunological and one might be specialized in, say, chromosome segregation. But here's the key in biology is the aggregation. It's a way, how do we take all these different studies and take the mean of that to have something better than each individual study? And I'm going to show you how we do that today. So why would I want to do this? Does anybody recognize this screenshot here? What is this? NCBI? Gene Expression Omnibus. This is where we're sticking our data. And right now, actually, this is a couple months. I think I took the screenshot a while ago. But it's probably, we now have about probably 100,000 different studies in geo at this moment. Okay? So there's a lot of crowds in there. So that's my goal is, how do I get this information out of here to actually impact my own research? And I use lots of tools. I'm going to show you those today.